Hello everyone, thanks for joining us today for the webinar that eMolecules is co-hosting with Solvay and Strem Chemicals. The presentation today is titled Phosphorus Specialties, the Cornerstone of Synthesis for Pharmaceutical Applications. My name is David James and I am the Senior Vice President for Product Management at eMolecules. So before we begin today, I just want to give a brief mention about what's new at eMolecules. We have actually just launched a new eMolecules offering where we can offer building block reformatting and reaction-ready vials. You can order gram, milligram, molar amounts of pre-weighed building blocks, reagents, and catalysts shipped in vials or plates. This can save you a lot of time weighing, and compounds arrive in the exact format you need for automated synthesis or parallel reactions. We can also incorporate the reformat button into our website, which then allows you to choose your reformatting options after you've already picked supplier provided pack sizes. This is completely customizable depending upon your vial type, the amounts that you want, and also what you would like us to do with the remainder, including storing in an inventory. If you would like more information, please feel free to contact me at david.james@emolecules.com. Okay, so today we have 3 guest speakers, all from Solvay, Dr. Dino Amoroso, Dr. Eamonn Conrad, and Dr. William Stibbs. Dino Amoroso obtained his BSc at McMaster University, his Ph.D. in the field of ruthenium catalyzed olefin metathesis at the University of Ottawa. Dino has spent most of his professional career working in the area of transition metal catalysis and ligand synthesis, focusing on catalyst ligand development and application. As a result, he has been involved in various industries, including electronics, polymers, materials, industrial chemicals, and pharma. His responsibility have included research, application development, business development, manufacturing, support, and sales. Currently, Dino is the North American account manager for Solvay's Phosphorus Specialties business. The next speaker will be then Eamonn. After completing a Ph.D. in phosphorus chemistry at Dalhousie University, Eamonn took up a postdoctoral fellowship at the University of British Columbia with a focus on polymer chemistry. Eamonn has since moved to industry, initially in a technical and project management role, to commercial and business development functions. Responsibilities include marketing, product development, production management, and general technology and commercial development activities. Currently, he is in Solvay's business development group and in Phosphorus Specialties, serving customers globally. And then the third speaker will be Bill Stibbs. After attaining a Ph.D. in organosilicon chemistry at the University of Sheffield, Bill moved to Canada to postdoc at the University of Western Ontario, where he focused on organosilicon and organogermanium polymers. Bill spent his career in fine and specialty chemical industry, primarily in the fields of metal, organic, and phosphorus chemistry. His responsibilities have included research, product development, small-scale manufacturing, application technology, product management, marketing, and business development. From time to time, he's even dabbled in sales. Currently, he is the business development manager for Solvay's Phosphorus Specialties business. Okay, so just a few rules. So just a few rules. To ensure a smooth running presentation, everyone is muted by default except for the speakers. There's going to be 2 ways that you can ask questions today. You can ask the questions yourself by using the raise your hand function, and I will unmute you. Let me know if you would like to ask your own question specifically through the questions box, or you can send the question through the questions box and I will ask on your behalf. Just let me know either way which one you prefer. The session will be recorded and available to all registrants. Any other questions, please feel free to contact me at david.james@emolecules.com. Okay, so that's going to be all from me. So now I will hand it over to Dino. Okay, should be over to you now, Dino.
Okay, great. Can everybody see my screen then?
Yep, looks great.
Yep. All right, great. Thanks very much, David. And thanks everybody for joining the webinar today. It's a great pleasure for us to have this opportunity to present a little bit about the Phosphorus Specialties business, but also focus in particular on some of the applications of those products in the pharmaceutical arena. So by way of an overview for today, we'll focus a little bit, very briefly, on who is Solvay's Phosphorus Specialties business. I'll also touch on Strem Chemicals and a new partnership that we've established with Strem for the distribution of the Phosphorus Specialties products. I'll give you an overview of basically how we make some of these phosphine ligands and phosphorus products in general. And then I'll turn it over to Eamonn and Bill, who will talk in more detail about the applications of those products in pharmaceutical catalysis, life sciences, and then of course we'll leave some time at the end for questions. So before we get into the Phosphorus Specialties business, I just want to talk a little bit about Strem Chemicals. I'm sure everybody on the webinar today is very familiar with Strem, having been established in 1964. They have over 55 years of experience in the manufacture and handling of high quality inorganics and organometallics. They have over 5,000 specialty chemicals available in their product listing. And of course, now that includes a range of phosphorus specialty products as well. Whether it's lab chemicals for research, cGMP products manufactured in kilolab suites, high pressure materials, custom synthesis projects, Strem has a range of products that suit a variety of applications. And of course, they have customers that span the ranges of academic, industrial, and government research labs, as well as commercial or industrial scale organizations operating in the pharmaceutical markets, electronics, chemical and petrochemical industries. And really this sort of broad reach that Strem has into research labs around the world was one of the main drivers for Solvay Phosphorus Specialties opting to partner with Strem for sample distribution. That, coupled with the emphasis on quality and know-how in terms of the handling of sensitive materials, makes Strem a perfect partner for the distribution of our products and facilitates those in the research community accessing those products very conveniently. So in terms of who is Phosphorus Specialties, for those who are unfamiliar, Solvay is a very large global organization made up of about a dozen or so global business units. Within one of those global business units, known as Technology Solutions, is where you'll find the Phosphorus Specialties business. So as the name suggests, we're a global supplier of differentiated products and technologies based on the chemistry of phosphorus. And so what makes Solvay's Phosphorus Specialties leader in phosphorus chemistry? Certainly, we have a long-standing history in innovation, over 160 years of know-how in phosphorus chemistry. Being a global organization, we pride ourselves on secure and consistent supply, so we follow very stringent quality control standards and leverage our global footprint to ensure that products are available around the world in a timely manner. We've always valued partnership. We've worked very closely with customers to translate their needs into concrete solutions and develop phosphorus-based solutions for those needs. We have exceptional scale-up capabilities. So whether you're working in research or pilot or industrial scale, Solvay and the Phosphorus Specialties Group have assets mirrored throughout that value chain in order to supply at any scale. And ultimately, it comes down to the phosphorus and the differentiated products that we offer. We have a wide range of phosphorus-based chemistries that find their way into a range of applications. So those differentiated phosphorus products, like I said, have a number of applications. And whether it's cylinderized phosphine gas that's used in agriculture as a fumigant or in electronics as a dopant, phosphonium salts that are used in biocide applications or as catalysts in the curing of epoxy products, really there's no limit to the applications of those phosphorus-based products. Of course, today we're going to focus on applications in life sciences and catalysis. So, as I said earlier, I'm going to give you a little bit of an overview of basically how we make some of these phosphorus products, and then we'll focus most of our attention on applications. So the chemistry that we're going to talk about today all begins with elemental phosphorus that you see in the top center of the screen here. While there's a lot of chemistry shown in the scheme below, we're going to focus primarily on the left side of the scheme. So moving from P4 over to phosphine gas. Solvay is the largest manufacturer globally of phosphine gas. We handle it at high pressures. And from that phosphine gas, we can make primary, secondary, and tertiary alkyl phosphines, which you see in the arrows directly below phosphine. We can derivatize those via oxidation or other chemistries to arrive at phosphonic or phosphinic acids, phosphine oxides, phosphonium salts, etc. Of course, we can also leverage the chemistry of sodium hypophosphite to arrive at things like phosphorus base acids, as well as phosphates and phosphonate esters and a range of other products, most of which you'll see in some of the examples we have today. So from phosphine gas, we need to find a way to make phosphorus carbon bonds. And one of the most practiced chemistries that we employ today is the free radical addition of phosphine to olefins. And so the schemes that are shown on the screen today are a relatively straightforward and pretty familiar set of radical-based chemistry. Essentially, we start with an initiator to generate a phosphine-based radical in that top reaction that you see. That phosphine radical can react with a terminal olefin, such as butene, that's shown in the middle reaction. Of course, through the propagation process of that radical chemistry, you generate the alkyl-centered radical. That alkyl-centered radical is quenched by the phosphine that's present, regenerating that phosphine radical and resulting in the formation of, in this case, monobutyl phosphine. Of course, if you were to repeat this process multiple times, eventually you would get to secondary and tertiary alkyl phosphines. And the scheme that I had shown with butene leads to, of course, tributyl phosphine, that's shown in the top left corner of the scheme here. This is a fairly standard product that the Phosphorus Specialties group offers, along with that trialkyl phosphine, and as a result of the sort of side reactions of the radical chemistry, that's of course accompanied by some isomers like secondary alkyl group that's shown in the top right. There are also some oligomers or telomers and some elimination products that are present in very small quantities. Of course, we can control the nature of the products based on the conditions that we use, and also we can manage the side products and impurities using some fairly traditional manufacturing or purification processes. So the tri-N-butyl phosphine is a relatively simple product based on a fairly simple olefin. When you start to think about the olefins that we can use in this radical chemistry, the structures can become quite complex and quite interesting as well. And so I've shown a sampling of some alpha olefins that could be used for this radical chemistry. On the far left are some relatively simple alpha olefins like ethylene and butene, which give those linear alkyl phosphines. Something like isobutylene in the radical chemistry will react to install an isobutyl fragment rather than an N-butyl fragment. These are all fairly standard chemistries and work very well under the radical conditions. Things like monounsaturated cyclic olefins also work very well to install a cycloaliphatic group like cyclopentyl and cyclohexyl. It gets a little more interesting when you have a diunsaturated or polyunsaturated cyclic olefin like cyclooctadiene shown at the bottom of that second column. We can install functionality like esters and amides and alcohols using functionalized olefins that are shown in the third column. And then, of course, there are things like aromatic olefins that, while certainly reactive, are a little less desirable because of the tendency of those things to polymerize under radical conditions. But really, the idea here is to emphasize the sort of range of structures that are available and the diversity of the chemistry that we have simply by practicing the radical chemistry. And so just to give you a flavor for some of the product diversity, I'll talk a little bit about the reaction of phosphine and cyclooctadiene. So I've shown in that scheme that phosphine can add to both double bonds in the cyclooctadiene structure, giving rise to a pair of isomers, one on the left that we call the asymmetric isomer and one on the right, which is the symmetrical isomer. And those are based upon how the phosphine adds across those two double bonds in the radical process. We sell this product through Strem as a mixture of isomers, Cytop 282T. Of course, recently Paul Pringle published a report on the separation of those isomers under using selective protonation, which is just another example of how the chemistry of phosphorus can be sort of refined to meet the needs of the application. Of course, we're not limited to radical catalyzed addition of phosphorus or phosphine to olefins. I'm showing here the acid catalyzed addition of phosphine to olefins. In this particular case, I've chosen to highlight the isobutene olefin. So strong acids will generate a carbocation at the most substituted carbon here. And of course, phosphine addition to that carbocation will lead to a slightly different structure than we saw for the radical addition. So under acid catalyzed conditions, phosphine adds to isobutene to give a tert-butyl structure, as you see in the bottom left, where you see di-T-butyl phosphine. Of course, under radical conditions, the product that you would isolate is a multi or mono-di or tri-isobutyl phosphine, shown on the bottom right of that screen. Interesting comment here. Based on the chemistry of acid catalyzed addition, tritert-butyl phosphine cannot be prepared under acid catalyst, but the tri-isobutyl phosphine can, and that's really comes down to a play on the sterics and the electronics that are involved in that chemistry. We also have other chemistries available to us. Things like Michael addition to activated olefins leads to some pretty interesting cyclic structures that you see on the top here. And then, of course, addition of phosphine to 2,4-pentane dione gives this polycyclic phosphoadamantane structure that you see on the bottom right, which is another product that is quite frequently used in catalysis as a building block for some interesting ligands and technologies. So that's basically a flavor for some of the synthetic chemistry that we use to arrive at the structures of our phosphorus products that of course, we're trying to highlight today. From here, I'll turn it over to my colleague, Eamonn, who'll focus more so on some applications in catalysis. Eamonn?
Thanks, Dino. Yeah, so as Dino mentioned, the next section here is going to be a bit around the specific applications and how our chemistry plays in that. Just move to the next slide, please. So for us, our main area where we focus on is the synthesis of these ligands, as outlined previously. You know, we have a variety of different systems that we're capable of, you know, achiral and chiral systems, monodentate, bidentate, what have you. And in a lot of cases, we're supporting these to catalyst manufacturers or end use, and it covers a variety of different reactions. So, you know, relevant today, I'm going to talk a bit about olefin metathesis, things like hydrogenations and hydroaminations, and how they're playing in pharmaceutical and life sciences. But we're not really limited to that. It's just really for the sake of time that we're just going to touch on a few items here. Next slide, please. So the interest in phosphorus catalysis is really driven around the diversity of the structure and the tunability of that structure, right? As Dino mentioned earlier, we can really tailor these systems to fit the needs that different applications are using and different chemistries require. From a practical standpoint, a lot of these are supplied as liquids for ease of handling, and we do a lot of product stewardship and safety around that. A key point as well is that, you know, we have established manufacturing routes to these products for large scale and small scale availability. So we're plenty capable of supporting these from an actual commercial standpoint as well, not just, you know, a gram here and a gram there. And ultimately, you know, we work with our end users to find that key balance between these phosphorus ligands and the metals that are being used. And we really are a leader in supplying these at commercial scale to the market safely. And with our partnership with Strem supporting the sampling and the research functions of it too, it's a really great combined effort now to get these into researchers' hands, and we hope everyone finds use from them today. Next slide. So the first topic I want to touch on is really ligand diversity, because that's really kind of the crux of a lot of catalysis. Without diversity and being able to tune the structure, you're really going to get nowhere fast. So what I wanted to start with was the example of the Josephos ligand family, because that's a really nice example where you've got this amine group in the precursor for the Josephos, and adding a secondary phosphine allows you to tailor that molecule quite dramatically. We've supplied several of these secondary phosphines ligands to the market. Highlighted on the bottom are just a sampling of 6. And being able to make all these different secondary phosphines allows you to make this breadth of this family, and that obviously translates over into other families of ligands as well. We're not just limited though to precursors, obviously. If you look at the case of diphenylphosphine, which is a product we supply as well, in our own cases, we'll actually convert that to much more complex and elegant ligands, such as the Rhodophos CP100R, which is more commonly known as Chirophos, and supply of those materials into different applications. Next slide, please. So the first reaction that I wanted to talk about was amino carbonylation, and this is a particularly challenging reaction when you're looking at ammonia as a raw material. You can have basically uncontrolled amination or deactivation, of the catalyst. What this example shows here is a palladium catalyzed reaction using the bulky adamantyl type ligand Cytop 292. So in this case, it's using ammonia and carbon monoxide. And what these products on the bottom show is that this system is actually quite amenable to a variety of different conditions and functional groups. So you've got alkyls and halides, and also different positions of halides. But further to that, this can branch off into thiophene chemistry as well. So it's got a bit more diversity within the chemistry available to it as well, supported by this ligand. Next slide, please. Carbon-carbon bond formation itself is obviously a critical aspect in a lot of pharmaceutical application, particularly when you're putting in a chiral system. The example we're going to talk about here is the synthesis of eptazosine, which is a commercially available analgesic. Particularly important to this is when you're making this molecule, you're generating a chiral quaternary carbon at the top of the molecule here. Just go to the next slide. And the way you do that is you actually do an intramolecular aryl cyanation from this nitrile species shown here on the left. This is done with a nickel catalyst and the Chirophos ligand, and also an aluminum-based Lewis acid. And in this particular case, you can do this cyclization, generate your chiral quaternary carbon, and also keep the cyano or the nitrile group in here as well, which is critical for the further chemistry to get to the final molecule. This is a really great example because the Chirophos shows you can get really high yields and really strong EEs as well, and that allows you to have really improved yields as you go through the process. Next slide, please. This ligand is also used in chiral hydrogenations, which is obviously a very large area for pharmaceuticals. The example I'm going to focus on here is around tolterodine, which is a treatment for overactive bladder disorder. And in this case, both isomers are active, but the R isomer is much more active and has more side effects, and so it's used in more extreme cases than the S isomer. So it's key to be able to synthesize these separately from one another. In this synthesis, you can see there's a Heck reaction for the starting materials here to generate your coumarin intermediate. And then the hydrogenation of that reaction is done with a rhodium-based catalyst and the Chirophos or CP100R ligand. In this case, it got really good 9:1 ratio of enantiomers. These can be separated through crystallization, and then these were carried forward to get excellent yield of the S derivative of the tolterodine molecule. Chiral hydrogenation has also been made famous by the DuPhos ligand family, particularly rhodium. And the reason for that is the rhodium DuPhos systems provide excellent rate enhancement and increased selectivity. And it also allows in a lot of cases to use a mixture of isomers in raw materials. The example we've got shown here is actually a chiral hydrogenation of an acrylonitrile, which is a reaction that's rare in its own right. And it actually goes into the formation of the molecule Lyrica, which is an anticonvulsant product that is sold commercially. You can see here, you start with a prochiral system, and you get this hydrogenation done with the methyl 2-phos, and you get really strong enantiomeric excesses of over, around 97% in these cases. Go to the next slide. Another famous reaction, I would say, of pharmaceutical and an important one is metathesis. This example here shows a key step in the synthesis of Simprovir, which is a drug first published by Janssen. In this particular case, this is a drug that's going into hepatitis C, so it's a direct-acting antiviral. And these are important because hepatitis C affects over 150 million people a year. And really in this ring closing metathesis step is critical because you generate this 14 carbon macrocycle. A variety of metathesis catalysts were looked at for this system, all ruthenium based with a combination of phosphine ligands and carbene ligands. And what was shown was that when using the tricyclohexylphosphine in combination with the carbene ligand IMES, it gave really strong improved space-time yields to form the product when the nitrogen is substituted with a Bach group. So they went through an optimization here and improved the synthesis quite dramatically by the addition of this phosphine ligand. Next slide. Another very large and important class of chemicals for pharmaceuticals and life sciences is indoles. They represent one of the largest structural classes in drug discovery. So there's always activity around new ways to do this type of synthesis. Highlighted on the bottom are just two examples here of commercially available materials that have indoles in them. So ondansetron is a nausea drug, and indomethacin is an anti-inflammatory drug. So not all routes are created equal to indole synthesis, so there's always a lot of work here, especially when doing substitution at the C5 position. Things like Fischer indole synthesis, which is obviously quite a famous reaction, is not always effective. So there's always new chemistry being developed with metal catalysts and phosphine ligands as an attractive solution here. Next slide. So in this example, they were looking at a one-pot synthesis of an indole. And in this particular case, so this is a titanium coupling reaction followed by an aza-Heck reaction. And in this particular one-pot synthesis, they found that the tricyclohexylphosphine gave much better conversions for the intramolecular cyclization step compared to carbenes, cheap phosphines like triphenylphosphine, which barely worked at all, and other monophosphine biaryl ligands as well in the presence of a base. So moving to the trialkyl phosphines in these cases really provided strong synthetic options to make these different indoles. Next slide. So the last application I'm going to talk about before handing it over to Bill for the life sciences is really about hydroquinones. And the reason I wanted to highlight this is because there's some very famous ligands that are used in a lot of pharmaceutical applications, such as things like Cataxium A. They're very useful because they have these bulky adamantyl groups on them, but they're also quite expensive, and in a lot of cases there's a lot of IP around them. We offer a product called Cytop 202, which is a similar molecule. So it has the bulky tert-butyl groups on it, and then the third substituent is the N-butylphosphine. And in certain cases, and this one shows here, is that it's as effective or more effective as some of these IP restrained, we'll call them, or more expensive ligands. And so there really are some viable options out there like this that can do these types of, say, ring forming chemistry and other chemistry that demands these types of ligands to provide real commercial solutions and provide a significant, not just yield, but cost advantage when we think about it commercially as well. Next slide. And so lastly, my last point that I wanted to make was, we talked about a few specific applications here, but we're really not limited to these as far as catalysis goes. We have a lot of diversity in our ligands possible. What the PNP ligand shows here, this is one that's used in olefin oligomerization and metathesis. There are cases where we do supply the catalyst directly. Rhodophoss NiCat is an example of that, where we make the nickel catalyst that can be used in olefin oligomerization and metathesis as well. And then also we can get these bulky structures as well, like the Cytop 292 here. and really anything in between. So if as interests come up and you have questions about different ligands, please don't hesitate to reach out. And with that, I'll pass it on to Bill, who will discuss life sciences.
Thank you, Eamonn. I'm going to go through a few examples, starting with Paxil. Paxil is an antidepressant that was originally developed by Glaxo. We like to show it as an example because there are multiple steps in its synthesis where phosphorus-based reagents can be used. So on this first slide, the initial step is showing the hydrogenation of the olefin, and this is catalyzed with an iridium catalyst with tricyclohexyl phosphine, so Crabtree's conditions used. The resulting alcohol is then converted through its reaction with sesamol under Mitsunobu conditions, and you can see we're showing triphenylphosphine is being used here. One option is to use trialkyl phosphines, such as tripropylphosphine, instead of the triphenylphosphine, which has the advantage of the byproducts being water soluble, which can make workup and isolation of the reaction product easier. Next slide, please, Dino. So in the total synthesis, this next step is described as the key step, and in this case, tributylphosphine is used as the catalyst for the alpha arylation using a bismuth-based reagent, and Dino is arylated regioselectively, and this compares well with complementary traditional palladium catalyst routes in that Koenig's base is used here as opposed to a strong base, and the regiospecific enolate generation is much higher than is obtained using palladium catalysis. Next slide. So in this slide, I show our first example of non-catalytic use of phosphorus-based reagents. So here we have a phosphorylation with DPCP, which is catalyzed by DMAP, and this enables the anti-selective copper-mediated allylic substitution using the chlorosilane, and the silane can then be reduced to the, oxidized to the allylic alcohol as shown. So in this case for Paxil, you can see there's at least 3 distinct steps where phosphorus-based reagents can play a role. So it's a very good example, and you'll notice that in all of these cases, phosphorus is not an element that's present in the final API. So later on, we'll see some examples where phosphorus is present. Next slide, Dino. So the phosphonium salt dual phos, so shown in the bottom left here, was developed by one of our academic collaborators, Jim McNulty at McMaster University. So using our triimpropylphosphine, he manufactured this phosphonium salt, and this is shown that this can be used to enable 2-carbon homologation of aldehydes to latent alkanals. So as illustrated here, the pinnacle protecting group on the phosphonium salt means that the other transformations can be performed easily to get to the end product, such as phosphomylides GNH, and then at the end of the reaction sequence, the protecting group is removed easily using the standard salt ferric chloride. Next slide. So tributylphosphine is used commercially in a process developed by Hoffman LaRoche to make Tamiflu, and it's used to reduce the azide to the imine in the reaction sequence shown here. This compares well with the conventional hydrogenation process using Raney-nickel. So fewer impurities are generated. There's no hydrogenation of the cyclohexene functionality, and the process to isolate the product is easier as no filtration is required. And there's no filtration required because the byproduct tributylphosphine oxide is water soluble, which means this simple phase separation process can be used rather than filtration. Next slide. So there are a number of ways that phosphorylation can be used in the manufacture of prodrugs. In the example here, you'll see that we have remdesivir, which I'm sure is an API that you've all heard a lot about in the last few months. So here, PDCP is used to generate a monophosphate, so a monophosphate nucleoside, which is the actual remdesivir structure. And this can be easily converted to the triphosphate metabolite, which in the body. So one of the problems in the body is the monophosphate generation is usually the slowest step to get to the triphosphate. So supplying the API as the monophosphate is a way to have a more effective drug substance. Next slide. So another example of prodrug use. In this case, this is fostemsavir, which is developed by V2IV and was recently FDA approved for treatment of HIV in adults where most of the treatments are no longer working. So in this case, originally temsavir, shown on the right, was developed, but for high doses, it was found that the temsavir is not soluble enough. So a synthetic route was developed to convert the temsavir to fostemsavir, which enables the higher dose to get to the required site in the body, and then that's converted back by the enzymes in the body to the actual temsavir. So here, to make the prodrug version, the temsavir is reacted with chloromethyl ditertiary butyl phosphate. So the product we sell is the ditertiary butyl potassium phosphate because the chloromethyl version is unstable and therefore cannot be supplied in a stable form, so it must be generated in situ from the potassium salt. Next slide. So the phosphoreagent shown at the bottom of the screen here is a high purity molecule that's used to make phosphoramidites of DNA and RNA bases. These phosphoramidites are then used to build up synthetic oligonucleotides. So the oligonucleotides can be used in diagnostics, for example, PCR and gene sequencing. From a therapeutic point of view, synthetic oligonucleotides have been in development for over 30 years, but so far less than 10 drugs have actually been approved, and the indications they've been approved for are generally small. Interestingly, at the moment, Novartis has submitted to both the FDA and the EMA an indication for inclisiran, which is an siRNA oligonucleotide for cholesterol reduction. And this has the potential to be a blockbuster drug, which is illustrated by the fact that Novartis purchased the medicines company for $9.7 billion, and they were the company that held the rights to inclisiran, and that was the main driving force for Novartis to make that acquisition. Next slide. So disulfide bond reduction is an important process in the gene sequencing tools. Phosphorus reagents are one of the reagents that can be used for disulfide bond reduction. So shown here is tris hydroxypropyl phosphine. And if you look at the table at the bottom with the model disulfide, you'll see that THPP and another phosphorus reagent, TCEP, with much better yields than an alternative diethiotrietol, DTT. So this gene sequencing technology is used in a number of ways. For example, around SARS-CoV-2, it was used to sequence the whole gene of the SARS virus. It can also be used to monitor mutations of the virus over time, so see how it's developing and altering its genetic structure. And the gene sequencing can also be used to track the origin of specific outbreaks. So for example, the outbreak in New York City back in the spring could be traced back to SARS-CoV-2 from Europe, whereas the initial outbreak on the west coast of the US could be traced to Chinese source SARS-CoV-2. So next slide. So to summarize, I hope we've shown that phosphorus chemistry has a wide range of applications in the life sciences. And if you're interested in phosphorus chemistry, we think we're a good company to work with because we're flexible and responsive. We're able and willing to innovate and look at new chemistries. And at the same time, we have large-scale manufacturing capacity and with the backing of a large corporation. So we have the support for logistics, registration, toxicological studies, etc. So thanks for your attention and participation in the webinar today, and we'd be happy to take any questions you might have.
Okay, thank you, Bill. So yeah, if anyone has any questions, please feel free to send them through the questions box. Or if you would like to ask your own question, I can unmute you and you can ask Solvay directly. So please feel free to send those through now. Okay, does anybody have any particular questions? Actually, here's a question from Stephen. I'll unmute you and you can ask your own question if you like. Okay, Stephen. No? Okay. If you want to send a question through, I can ask on your behalf as well. Okay, we have a couple of other questions here, guys. So one question is, how much flexibility do you have in making biphosphenes based on the 1,2-phenylene backbone? I'll let you guys decide.
Yeah, so I can take that one. So in short, a lot. We have basically a specialty group in one of our facilities in the UK. That's an area of focus for them. They've been doing that chemistry for well over 50 years, and they have special kit for it as well for both the chiral and achiral versions. And if there's any specific molecules they want to discuss, we can happy to talk about those ones offline.
Okay. Great. So then another question here is, any updates or importance on NP ligands? NP ligands, rather simple P ligands.
So maybe I can comment on that. It's Dino. So yeah, certainly NP or PN ligands are highly valuable, highly useful class of ligands. I think in our presentation there was really only one example of something like that, which was the Rhodophosph PNP. In general, we don't necessarily list a lot of those products in our product catalog, but those are certainly motifs that are available to us with the assets and the chemistries that we practice. So while we don't necessarily have a number of too many examples to highlight today, that is chemistry that's available to us.
Yeah, and I think that's, just to jump on that, I think that's a key point is we present a lot of stuff that's in the public space, but there are a lot of projects that we engage with specifically as one-on-ones and specific systems for specific end users. So, while we talk about some chemistry, this is certainly not, this is ultimately the short list of what we do.
Okay. So another question here is, how pyrophoric is tributylphosphine? How can you improve its safety?
So maybe I'll start on that one. So tri-N-butylphosphine is listed as a pyrophoric material. So formally speaking, it's either pyrophoric or it's not, based on a couple of different definitions. But certainly there are levels of pyrophoricity that many of us are familiar with from handling materials. Tri-N-butylphosphine itself is certainly pyrophoric. It will smolder if it's exposed to the atmosphere. So smoke, certainly. If it comes in contact with things like paper, like Kim wipes, or even things like finely divided solids, it can certainly ignite. We do have a few options, though, available to us to mitigate the sort of complexity that the pyrophoricity brings with it. First and foremost, we can, we have specialized cylinders that are designed for basically airless or air-free transfers, whether it's within our own facility or in labs and industrial settings. We have specialized cylinders that can connect air-free to ensure there's never any exposure to air. And so neat tributylphosphine, even though it's pyrophoric, can be handled very safely, very comfortable in its neat form. That said, we have a couple other options at our disposal as well. We have and can supply the product diluted. So in diluents like toluene or isopropanol, we can render the solution non-pyrophoric, typically around 50% dilution by weight. Things like trialkylphosphines are no longer pyrophoric, and so those are still very highly concentrated solutions that make them very efficient in use, but also eliminates the risk of pyrophoricity. There's also the option of quaternizing those alkyl phosphines, so we can make the phosphonium salts, which are air stable, certainly no longer pyrophoric and no longer susceptible to any sort of oxidation. And then so when it's time to use those, they're basically neutralized with a base, and you liberate the free phosphine. So there's a number of options available for the safe handling of pyrophorics like the tri-N-butylphosphine.
Okay, great. Okay, we have someone that will ask a question. Stephen, I can unmute you now and you can ask your question. Okay. Stephen, you should be unmuted. Oh, I guess we still have some difficulty there. Hello, Stephen? Okay, I can ask his question on his behalf. His question was, what types of phosphorus molecules were difficult to make and where research could be done on the academic level?
That's a loaded question. Maybe I'll start, and Eamonn and Bill, you can jump in with your perspective. So the chemistry of phosphorus is very complex, and we focused on a really narrow subset of that chemistry. I think where the challenges remain, certainly, in identifying new motifs, more complex structures is something that you see in the literature ongoing today. People are still developing new ligand structures, more complex. I think from our perspective, developing simple and efficient synthetic routes to some of those complex structures is really key. In many cases, those very elegant, very complex structures are really not commercially viable because of the resulting cost associated with the complex syntheses that are required. And so I would say developing straightforward chemistries like the radical catalyzed addition chemistry that I presented, things that are very simple and won't add exorbitant amount of cost to those products, ultimately makes the phosphorus technologies more viable. And so for us, simple synthetic routes to complex structures is always going to be an important area of focus. I don't know, Bill or Eamon, if you have some other thoughts on that.
Yeah, I mean, I think thinking about catalysis specifically, there are many examples in the literature of very specific ligands that perform very specific transformations in better ways than existing simpler ligands. The challenge, as you said, is always how do you make those ligands economically viable at commercial scale. So again, it's more around synthetic routes, I would suggest, rather than developing new molecules.
Yeah, I was gonna say from my perspective is really a couple things. It's really, like you said, that simplicity and complexity, especially when you get to like asymmetric chiral bidentate ligands and these types of systems, there's usually a lot of steps that goes into making them. Like when you see like a 10-step synthesis, that's not really going to be economically relevant in a lot of cases. But the other aspect too is also purification and, improving the yields and antimeric excesses of some of these wall is always something of interest. 50% yield can be sufficient sometimes, but if it gets up to 70, 80 in some of these complex systems, through hook or by crook, those are real game changers in a lot of these applications.
Okay. So we've got some more questions here. Can you reuse the used catalyst for the next batch? Can you recycle the catalyst in plant scale? Or I guess like which ones could be recycled?
So I'll take a first stab at that. So ultimately that's process dependent. The short answer is yes. When you look at some of these, especially things like rhodium in them, there's a lot of efforts at different companies to recover the metal and reuse those because of the expense of them. There's more interest in ones like cobalt catalyzed as well too, because cobalt's becoming a more important metal and in-demand metal with the electrification of the world. As far as the ligands go, I mean that's one you want to talk about an area that could use some work. In short, some cases there is, but some of these ligands like the DuPhos ligands, if there's viable routes to recover them, a lot of people keep those proprietary in nature, but any developments around there are certainly welcome, since the literature, to be quite honest, in the public space is minimal.
I think one of the challenges is that often the precious metal is the focus of recycling, and whatever process is used to recycle your precious metal destroys the ligand. So ligand recycling is not always viable. I think there are small scale efforts to recycle ligands around things like ligands supported on zeolites or silica, that kind of thing, where you have you're almost heterogeneizing the homogeneous catalysis system. So that's one way that you make reusability, I guess, rather than recycling of the ligand possible.
Right, and just to add to that, Bill, yeah, so the metal's generally the focus when it comes to catalyst reuse or recovery. There's certainly interest in the, let's say, phosphorus chemistry community around phosphorus recovery, and that's something that every year gathers a little bit more interest. One sort of unique case is, and somewhat relevant to recovery of the phosphorus, is around the use of things like tributylphosphine in the Tamiflu synthesis that you highlighted, Bill, where you have the ability to extract the product and manage it quite simply. So compared to something like triphenylphosphine used in in that process, the alkyl phosphines, whether butyl or N-propyl, are used in smaller quantities because it's not a solid. It's much easier to handle, and the activity leads to a dramatic reduction in certainly in the volume of material like waste product, but also in just the sheer amount on a molar basis because of the sort of advantageous nature of the alkyl phosphine over a aryl phosphine. So in short, you know, focusing on optimizing the chemistry of the phosphorus component can also help to not necessarily recover catalysts, but at least minimize wastes and that sort of thing, and make the processes a little bit more sustainable.
Okay, so next question we have here is, do you offer or plan to manufacture Denton's catalytic Mitsunobu reagent?
But I'm going to say, as we all have blank faces, the answer is obviously no, not at this point. But you know, as Eamonn already said, if you have specific molecules of interest that we don't list, we'd like to learn more about the opportunity associated with them and what your interest is in them.
Yeah, I was going to say, you got two business development guys on this webinar. Sounds like a perfect conversation to have with those two guys.
Yeah, it's one we'd be happy to entertain. So right now the short answer is no, but it's more because there's of the demand. We haven't been specifically asked, I would say, at this point. Yeah, ultimately, I mean, one point of that is, you know, we're not a catalog phosphorus company at the end of the day. You know, we rely on companies like Strem who focus really in that area and providing that breadth of materials. We have some, and we offer them through Strem. But the reality is a lot of these specific ones, when they become a specific commercial need, we will work with the end users around developing and scaling up those activities.
Okay, great. Okay. Unless anyone has any last questions, I think we covered everything. Okay, great. Well, thank you all for the very interesting talk. If anyone has any additional questions, please feel free to reply to the registration email or myself at david.james@eMolecules.com. We may also have the opportunity to have some follow-up sessions on specific topics. So if you want more information or details, please let us know and we can invite you to those as well. All right, Dino, Bill, and Eamonn, I thank you again today for the presenting today. I thank everybody in the audience for listening, and we wish you all a good day or good night. Thank you very much, everyone.
Thank you, David. Thanks, everybody.
Thanks, everybody.
Thank you.